Good afternoon, everyone, and thank you for Dr. Go and INBIOSIS for allowing me to do this presentation and sharing today. So basically, what I'm going to talk about is the RNA-seq application in MGI. So as Dr. Go mentioned, MGI is a subsidiary of BGI, and I'm the field application scientist that's based in Malaysia. So yeah, I will introduce to you more about RNA-seq applications now. So yeah, these are the contents of my talk today. Will be a little bit of introduction, then some applications, and of course our MGI RNA-seq solution using our proprietary DNB-seq technology. So first we talk about RNA sequencing, the introductions and applications. So I think we are all aware, or I believe all of us should know by now that RNA-seq is a tool for measuring expression across the transcriptome. So sometimes RNA-seq, people refer to it as whole transcriptome sequencing. So mainly it's a quantitative method to determine the absolute quantitative value of each molecule in a cell population. And with this information, we can directly compare the results between experiments. So common setup for this kind of experiments would be a treated sample and a non-treated sample, or maybe a disease sample versus a non-disease sample. So we can compare the gene expression between these two groups, and then we can infer some biological results from it. So when we talk about RNA-seq, these are the three key points or keywords that we normally hear or refer to. First is quantify, second is discover, and then third is profile. So all these components or all these keywords will include like transcript isoforms, gene fusion, single nucleotide variants, allele-specific or gene expression, and other features. So these are some of the applications in RNA-seq. So basically it's divided into two areas for me. So first will be profiling, where it includes differential gene expression analysis. As I mentioned just now, you have your treated sample and non-treated sample. So you compare the different expression between these two groups of samples, and then you see which set of genes is upregulated and which set is downregulated. And then with this information, it can lead to like development of biomarkers and SNP detection as well. So as for the discovery part, usually the basic experiment that we will do is from the DEG analysis. So from there, we can discover some novel transcripts, gene fusions, exons, or alternative splicing and all this. So when it comes to gene expression analysis or differential gene expression analysis, and also the transcriptional profiling, these two applications, we are basing it on a very general assumption that whatever changes in the transcriptome or the mRNA levels, this will correlate to the phenotype or the protein expression in that particular biological system or sample that you are sequencing. So other than that, it will be the identification of splice variants, and as I mentioned, novel gene identification. And some studies are actually interested in just the overall look at the transcriptome. So they just do transcriptome assembly to see what are the genes that are being expressed in an organism compared to the genome. Because I think, especially for eukaryotes, not all the genes that is in the gDNA is transcribed. So we just look at the transcriptome, and you can just do the assembly to look at the overall picture before further formulating your biological questions or experimental objectives. And then, of course, lastly is the SNP finding. So there are many types of RNA sequencing applications, and as I believe we are all aware that RNA composed of different types of RNA. So the most common one that we study is the mRNA. So from mRNA, we can do the de novo analysis or reference-based analysis, and of course the RNA-seq. Then the other part of RNA is the non-coding RNA that researchers often look at, which consists of small RNA, long non-coding RNA, and circular RNA. So this figure here shows some of the basic applications for RNA-seq. So first is the transcriptome sequencing. So basically, we take the mRNA and study the expression and structural analysis, and of course, some sequencing assembly and etc. You can do quite a number of things with your mRNA. And then for whole transcriptome sequencing, so the difference between whole transcriptome and transcriptome is actually the mRNA and other RNA. So whole transcriptome means you will include other types of RNA. So, but then essentially the research content is similar to transcriptome sequencing. So you want to look at the expression and function of all the respective genes in that organism. And then you have small RNAs. So small RNAs are usually less than 200 base pair. They are quite short and they are not involved in any of the expression, but some of them do regulate the mRNA expression or promoter regions and all that. So by sequencing the small RNA, you can also do some expression and functional cluster analysis of small RNAs. And then there's another type of application called RNA capture sequencing, where we sequence the RNA exon regions. So especially this is quite common in severely degraded FFPE samples, where it's very hard to get the gDNA sample or even very hard to get any nucleic acids at all. So some people will opt for this kind of route. And yeah, the research content is similar as the other. So it's for expression and functional cluster analysis for RNA coding regions. So now I will share with you some examples of RNA-seq applications based on projects that is undertaken by BGI and MGI and using our DNB-seq platform. So the number one most common application, at least for in the research community, is the differential gene expression, as I mentioned. So in this project, basically, vitellogenin is a protein that is involved in the egg yolk production in this organism eel. So they use they compared the transcriptome of pre-vitellogenic samples and early vitellogenic samples of ovaries from wild-caught eels. So basically, they use this to study the expression of specific genes that's encoding the cell junction proteins that could be involved in regulating the protein uptake. So what they found is that from the gene expression patterns, it helps to identify suitable candidates that's involved in the regulation of VTG uptake, and also provided novel sequence data for the eel, including putative receptors corresponding to LRA and LRP13 members of the low-density lipoprotein receptor family. So another popular example, I would say, is in the infectious disease landscape. So this is a study based on the global gene expression profiling of SARS-CoV virus. So this is a virus affecting swine, affecting the animals' pig. So basically, they took a non-infected sample with an infected sample, and then they sample at different time points for the experiment. So this is a Western blot results showing the expression at different time points and the negative and positive samples. So you can see that the virus is actually not present in the negative samples, and then it's slowly increasing in load from as the time goes by. And then GATPH is the internal control for Western blot usually. That's what we do. So they sequenced the RNA of both the samples, and then they did some cluster analysis, and then they try to map out the transcriptomic landscape of this swine infection in pigs. And then next, this is quite a new topic in Malaysia, I believe, but it is quite used widely overseas now. So the term is called dual RNA-seq, where researchers actually sequence both the pathogens RNA, mRNA, and the host mRNA. So they can see what are the genes that's interacting with each other during an infection. But this paper is an example of the interaction between a bioeffector of this and the pathogen of the ginger plant. So basically, in their experiments, this bioeffector, it can inhibit the growth of this pathogen, but then they are not sure how it works on the molecular level or on the biological level. So they sequence the mRNA of these two organisms, and then they compare the gene expressions that's involved in certain pathways. Like here, this is the gene ontology term. So some are upregulated and some genes are downregulated. And then you can see that for our RNA-seq experiment, so this is done on the DNB-seq, the G400. It is one of our mid-range sequences. So when they do the QPSR validation, you can see the results are tally with each other. Yep, and then this is a very recent paper, I think just released a few days ago or a few weeks ago. So this is the first ever domestic pig cell gene expression landscape. So if you recall, I mentioned earlier in the presentation that not necessary we just compare sample 1 and sample 2 and compare the expression. Sometimes we would like to know what is the overall picture of the gene expression in a particular biological system. So here they sequence of 200,000 pig cells from 20 different tissues and organs from the pig, and then they characterize the heterogeneity of cells and identified over 200 cell clusters, which represents 58 major cell types. And then they also perform some integrative analysis of endothelial cells, which reveals a high degree of heterogeneity. And then, of course, you can use this information to identify several functionally distinct endothelial cell phenotypes for whatever purpose of the experiment or your research is. And then some of the findings of this project is that the intercellular communication analysis predicts that the tissue and cell type specific crosstalk between endothelial cells and other cells. And then the Regulon analysis of single cell transcriptome of microglia in pig and 12 other species also further identifies that this gene is evolutionary conserved Regulon in the microglia. So basically, this whole project is a single cell sequencing transcriptome, and they are using our DNB eLab C4, which is a single cell sequencing machine or device. And then this is our DNB-seq T7, which is one of the higher throughput sequences that we are carrying. So other than that, RNA-seq can also be applied in spatial transcriptomics. I think this has been the talk of the town for a number of years recently. So basically, spatial transcriptomics is like drawing a map of the biological interaction or or molecular mechanisms that's happening in a particular biological system. So one of our customers, INBIOSIS in Singapore, they actually purchased one of our biggest sequences to do this spatial transcriptomics. And because usually we require a huge amount of data, so this machine in the background here, if you can see, it occupies a whole room, and the throughput is about 72 terabytes of data per run. So this company, they use this technology to discover biomarkers for disease, early detection, and precision medicine. So this is our using our DNB-seq platform. Okay, so now I will move on to introducing MGI as a company and our proprietary DNB-seq technology. So MGI's main mission is to develop and promote advanced life science tools for the future of healthcare. And we do this by providing real-time, complete and lifelong life science solutions to our customers and researchers throughout the world. And in the recent years, we have become one of the three big suppliers globally to actually mass produce proprietary clinical genetic sequences. So our headquarters is in China, Shenzhen, but now we have expanded globally worldwide. So like me, I'm based in Malaysia. So yeah. Okay, so our main business segments can be divided into three parts. So first is the genetic sequences, second is laboratory automation system, and we have some miscellaneous new products that is listed here. So just to briefly go through each of these three segments is that our genetic sequences, we have the lower throughput to the highest throughput. So this is the G50 is a low throughput sequencer, G400 is medium, and T7 is a high throughput sequencer. And then this TX is the even higher than the T7. And of course, with our genetic sequences, we do provide solutions for sample extraction to library preparation and all that. And then when it comes to our lab in automation system, we have systems for sample preparation where this is usually used for clinical setting where we have samples in like different tube sizes. So these systems actually transfer the samples from that sample collection tube into 96 well plates without having any hands-on manual hands-on. So this can actually save you some biological hazard in handling potentially dangerous biological samples. And then secondly, we have an automation system for automated nucleic acid extraction. So this is developed due to the COVID-19 pandemic. So a lot of customers are actually using this to extract the nucleic acids from the COVID-19 samples and then later used for either qPCR testing or sequencing. And then we have our automated liquid handling workstation. So we have the MGI-SP 100 and 960. So these instruments are actually used to prepare our DNA library or RNA library. So these instruments, another advantage is that it can also perform nucleic acid extraction, whereas this one actually just do the extraction. This instrument just can do both extraction and library preparation. And of course, we have our proprietary kits for all these instruments. Then others include remote robotic ultrasound system, the DNB eLab C4 that I mentioned earlier. So this is our pocket single cell lab. It's about the size of a USB pen drive. And then we do have bioinformatics related products and also a mobile laboratory platform. Okay, so just want to mention a little bit of our DNBC workflow. So you can see the first step is fragmentation because MGI's platform is still using short read sequencing similar to Illumina. So we need to fragment our DNA or RNA sample into smaller fragments, then perform the size selection to select the desired size that we are interested in, and then followed by end repair, A-tailing, adapter ligation, and PCR. So these steps are quite common across all the sequencing platforms, but what is unique to MGI is actually this part here, the circularization, where we take our double-stranded DNA library and make it into a circular library. So I will explain more in the subsequent slides. And then, of course, this workflow is quite common among sequencing or NGS platforms. So we have to do some maintenance on the sequencer and then do some preparation before we can run the sequencing. Okay, so for RNA-seq workflow, this is an overview, and basically you notice some of the steps that I've shown in the previous slide is actually quite similar to this. So basically, when you extract your RNA from your sample, you will have your total RNA. And if you are interested in looking at the gene expression, so you have to get the mRNA sample, mRNA part from your total RNA, because especially for the the rRNA, it will be very abundant in any given sample. So we have to either do a mRNA enrichment to get your mRNA from the total RNA, or another step you can do is the rRNA depletion, whereby you just deplete the rRNA from the total RNA. So there are few differences between these two methods, which I will not go through in detail in this slide. So basically, just wanted to show you the overall workflow for now. So mRNA, and then you have to do your reverse transcription into cDNA, and then perform the second strand synthesis, and then followed by the usual end repair and A-tailing, and then adapter ligation and PCR. So after PCR, you will get your double-stranded cDNA library here, where we need to heat denature the double strand into single strand, and then we add the spleen oligos and all the buffer and enzymes to make it into a circular DNA. So this process is called circularization, and it is the first step in our DNBC technology. So this is unique to MGI. So yeah, this is basically the structure of the library that we have. You have your insert fragment here with some adapter and barcode information. So this barcode is for sample multiplexing, basically. So heat denature, you get your single strand, and then you add the spleen oligo and all the other components to make this single strand into a circular strand library. And after you get your single strand, we need to make the DNB. So DNB basically stands for DNA nanoball. So DNBC is DNA nanoball sequencing. So the way we make our DNA nanoball is through a process called rolling circle amplification. So I have a video here that show you how it works. So first we denature to single strand and make it into circular DNA. And then we have some polymerase here to do the extension of your fragment. And then it will displace the original strand that is there. So this process will repeat between 300 to 500 times. So basically, you have one very long strand of library, which will curl up into a DNA nanoball. So you can imagine it like it's a ball of yarn with rolled up. So all the fragment, all your DNA is rolled up into a DNA nanoball. So that's why we call it a DNA nanoball. And some of the features of the DNA nanoball is it is a linear amplification. So unlike PCR is an exponential amplification where 1 become 2, then 2 become 4, 4 becomes 16 copies. So for rolling circle amplification, it's linear. So 1 copy become 2 copies, become 3, become 4 copies. So because of this linear amplification, the amplification bias is low, and there's no error accumulation during this whole process. So you can imagine PCR, if there's error in one of the steps, you amplify it by 2 to the power of n times. So that is the meaning of error accumulation. Whereas for linear amplification, you just maybe one error will just carry forward to the next one, but then you are still amplifying the original strand. So the mistake would not be carried forward. And because it's a DNA nanoball, so the index hopping is much lower compared to the other platforms. So some of the people may ask, why is making DNB important, or why do we come up with this kind of technology why do we go through the trouble of making a single copy into multiple copies? Well, you can imagine it like this. So if you have a single strand of your template or your interested region, you can just imagine it as a single matchstick. And if you light up the matchstick, the light produced by a single matchstick is not really dark, even in a dark room. So if you have a DNB, because we amplified the original strand to between 300 to 500 copies, so you can imagine we have about 300 to 500 matchsticks, and if you light up all of them together, the signal will be much brighter. So you can easily differentiate the actual signals and the background noise. So after making the DNBs, we come to the third part of our DNB technology, which is the pattern array flow cell. So we have our DNB mixture here, and we need to load into the flow cell. And if you look into the flow cell, you will see this dot matrix, which we call a pattern array. So basically, each of the DNBs, it will be attached to each of the spots on the flow cell. So they have specific diameter and specific distance between each other, depending on which flow cell we are using. So they are attracted to the flow cell through electrostatic interaction. So basically, the DNBs are negatively charged, while the spots on the flow cell is positively charged. And then after that, we can perform the sequencing. So our sequencing technology is called the combinatorial probe anchor synthesis technology, or CPAS for short. And this is basically the process. So we will need to do a pre-treatment before sequencing can actually start. But after that, when the sequencing starts, it will incorporate fluorescent DNTPs onto your DNA nanoball. So, and then a light will shine on the flow cell, and then the excited fluorescence from the DNTPs will light up. So our imaging system or our camera system will capture the image on the flow cell, and these images will be used for base calling. And after they successfully capture the image, so the DNTPs, the fluorescence will cleave away from the DNA nanoball, and then it will repeat again this cycle for the second base in the sequence. So basically, this whole process will repeat depending on what kind of sequencing mode you're using. So for example, if you're using PE 150, so this cycle will repeat for 300 times. Or if you're using SE 50, so this cycle will repeat 50 times. And then finally, we have to do some maintenance on the instrument, so which is cleaning. Okay, so this is just a summary video of the workflow for DNB-seq technology. So this is our flow cell where you need to load the DNB. And then under the microscope, you can see this is how it looks like. And then this is the imaging during the sequencing. So each of these DNBs will be emitting a light that corresponds to the different bases. So after the image is captured, then we can use it to call these bases. Okay, so this is basically a short introduction of our sequences. So as I mentioned, we have our T7, G400, and G50, and you can see the maximum throughput per run listed here. So this is a very high throughput, 60 terabytes of data you can obtain from the T7, whereas G400 maximum you can get 1.4 terabytes, and G50 is around 150 gigabytes. So if you want to compare with Illumina, this is like similar to a MySeq. This is a HiSeq, and then this is the NovaSeq. And the average running time is slightly faster compared to the other platforms, and we do support a wide range of read length from SE50 to PE200. And this year we are actually launching the PE300 kit for the G400 sequencer. So this is our total solution for RNA sequencing from MGI. So depending on what application you're interested in, we can recommend different kits and different sequencing protocols or sequencing modes, either SE or PE. So we do have rRNA depletion kit and also the RNA library prep kit. We do have the normal universal RNA library prep kit and also the directional RNA library prep kit. So if you are looking at specific targets, like for example, the recent pandemic, the SARS-CoV-2 virus, so we know that the virus is an RNA virus. We can also use RNA-seq to sequence this virus, and our kit is called the Atoplex RNA library prep set. So the principle is slightly different compared to this, but I will not go through in detail here. And then for bioinformatics analysis, we have our MegaBot bioinformatics analysis hardware or workstation. It also couples as a server. So just to put into perspective, a normal workstation can maybe analyze your sample. We are talking about whole genome human sample, for example, at 30x coverage. So one sample is about 100 gigabases. So on a normal workstation, you probably need about 1 to 2 days to process your whole genome human sample. But on MegaBot, it can actually analyze the basic analysis in 2 hours, 2 to 4 hours. So because this MegaBot actually contains a hardware accelerator to speed up the bioinformatics analysis process. And then for specific applications like the SARS-CoV-2, we do have specific softwares to perform the assembly and do the analysis for pathogens. So basically, this is the end of my talk. So today we went through a little bit on the RNA-seq applications, gene expression profiling being the most commonly used application for RNA-seq. And then secondly, from this profiling, we can also discover novel genes. And of course, all of this is actually used to study the biological pathways and molecular mechanisms. So whatever method you use, I think our end goal for RNA-seq is always to understand what is going on the biological level and on the molecular level. So this can be widely applied in infectious disease and precision medicine. So example is the COVID-19 virus, and precision medicine is mostly in cancer or in genetic related diseases. And then we also talk a little bit about the MGI DNB-seq technology. So I introduced some sequencers and automation systems. And the take-home message from this is our system has increased accuracy because of the generation of DNA nanoballs. And the duplication value from the sequencing is also low, as well as the index hopping. So with our systems, we do have some library and sequencing kits depending on your needs. So that's all for my talk today.